name is Rahul Jain and I'm a postdoc researcher in the MiCRop project. I recently joined the MiCRop around like 8 months before, and I'm being a microbiologist by training. I'm looking into how the microbes in the soil, they interact with the plant roots.
In the greenhouse, we have two types of plant. So one is the normal plant that we grow, while another plant could be any kind of stress plant. So in my case, what I study is nitrogen stress. But it's not just nitrogen, but the stress could be either phosphorus or attack with a pathogen. And these two circumstances we study to see how the plant roots has different microbes colonizing around it. So to do that, we extract the DNA from the soil and we load it onto this machine. So this is a sequencing device which can sequence or which can decode the information in the DNA. And after this sequencing is done, we perform it, we analyze some of the data analysis on the computer using some bioinformatics tools to see which are more abundant under stress condition as compared to the normal condition. This is the soil that I've collected from the plant, let's say tomato, and I want to study what microbes are in there in the soil and how they help the plant to grow better. So first step for extracting those microbes are to mix the soil with sterile water. So I've already mixed the soil with the water, and after this what we do is we take a culture media. So this culture media that I have it has enough nutrients, or it has all the food materials or food ingredients that a microbe need for its growth. So what we do is we take some of this soil slurry that we made, we mix it, we plate it on the nutrient plate that I showed you, and then we spread it evenly on the surface of this nutrient media plate. So as I said, it has enough nutrients for the microbes to grow. Next step is to just put them in a optimal condition. So here we use an incubator to do that. So these microbes, in let's say in 5 days or 6 days, they can grow, they can form small colonies which we can isolate them. Just to show you that how it looks like after 5 days or 6 days of growth. So this is one of the bacteria that I isolated from the soil that I've showed you. So when we grow the plant in the greenhouse, so we want to understand how the plant is behaving, and in the soil how the microbes are being recruited. So if you see this graph, to understand the behavior of the plant when they grow under normal condition and when they grow under any nutrient starved condition. So you see after 2 weeks, after 1 week, there is like a variation in the biomass of the plant or the weight of the plant. So there is more biomass when they were given normal or every nutrient, but they were depleted with any kind of nutrient, there was less biomass. And then we wanted to move that how bacteria, they are going up and down when they were stressed. So you see clearly, these are the stressed plant, these are the control plant. And the microbes, you can see here, the microbes in here, there are few microbes which are only present in stress condition but not in control condition. So in another example, if you see, again these are the stressed plant, the red ones, and the blue ones are the normal plants. And you see after 1 week and 2 weeks of the stress, you see these bacteria, some of these bacteria, they are more in number in the stressed condition as compared to the normal or the control condition. So this indicate that these bacteria has some role to play. They probably help the plant to grow better when there is stress. And this data we use to isolate the microbes and see or study how they function, how they interact with the plant, and this information is really critical for us to do further research on the grounds.